Today what I'd like to do is talk about, as Cameron says, the nucleotide metabolism of Schistosoma Mansoni. This is very much a UK-Brazil collaborative project. And in a way, what I'd like to do is start with, on the first slide, is acknowledging the sort of large number of people. The project was driven by Humberto in Brazil, and a lot of the work has been carried out by his two graduate students, with contributions from the people at Diamond Light Source and then us in the PPF. So what I'd like to do first is sort of just remind those of you who don't know what schistosomiasis is. So it's a human trematode flatworm. It's one of the causative agents in schistosomiasis, which is a disease of poverty, and according to the WHO, in socioeconomic terms, is second only to malaria in its effect in tropical countries. So its control is really based on one drug treatment. So as you know, we've seen the common theme that if there starts to become increasing resistance, then you have a problem. So what I'm going to focus on today is the purine selfish pathway, because the worm is actually totally dependent on the purine selfish pathway in order to supply the purine precursors it needs for its energy and its polynucleotide biosynthetic needs. So as such, as an essential pathway, it represents a target for novel drugs. So the aim of this project was to structurally and biochemically characterize the nucleotide metabolism of Schistosoma Mansoni. So this is what we do in the OPPF. We're a national resource centre for protein production. We're a service where it's free at the point of access for UK academics, and there's access to Europeans through the Instruct funding mechanism. So what we do is we sort of parallelise the first stages of protein production, so the cloning, the small-scale expression screening, and then to a certain extent also the protein purification and the crystallisation as well. So in terms of this project, it's been very much a sort of collaboration. So the target entry in the bioinformatics were done sort of by us and by collaboration within Brazil. So there was a lot of toing and froing of emails between myself and Humberto about the experiment that we were going to do. And then just about 2 years ago now, we did our first pilot experiment where it was 48 constructs through. So I did that myself. I took those through and then we reported the results back. And that was, in fact, extremely successful. So then Humberto's two graduate students then came over and carried on, did a larger scale experiment. And then the protein purification, some of it happens in the UK, a lot of it happens in Brazil. Crystallization happens in the UK and Brazil, and the crystals are then shipped largely to the UK for data collection. And then the structures are then solved back in Brazil. So there's a lot of toing and froing. And so the idea is that we kind of parallelize the very beginning of the process so that we sort of rapidly explore expression space to arrive at proteins for crystallizations. And actually in terms of our proximity, we're immediately adjacent to Diamond Light Source, so there's a lot of synergy in terms of our interactions. So it's sort of a very, very brief overview. We had 23 targets at the beginning. Those of you who counted the number of enzymes on the original slide, there are actually 14. So we've actually looked at some of the enzymes involved in pyrimidine synthesis and salvage, but also a target. If you use a codon, we've also explored codon optimization for one or two targets. So a target is just a different DNA sequence. So we've designed a total of 128 constructs. We made, if you average it out, 5.6 constructs per target. So a construct is a combination of a tag and a coding sequence. So for some of the ones that we knew would be more difficult, and there was sort of information from the bioinformatics, perhaps made truncations, we did fusion partners. We always tag with a polyhistidine tag, so an N and a C-terminal tag. I haven't got a pointer, but if you look here, this is a group of 5 constructs. So we have a C-his tag, an N-his tag, there's a sumo fusion there, a TRX fusion, and then an MBP fusion. So we just go all out at the beginning and it depends on the project, but more than one project, a target or construct per target. So of those 128 constructs we initially designed, we made 125. 69 of them expressed in E. coli. And the number of targets that actually expressed was 19 overall. So 19 out of 23 targets we had protein expression for, and those targets were then put into crystallization. And Josie is going to talk about the sort of outcome of the structural studies.
Thank you, Louise. Yeah, I'll take from Louise at this point. Learning to drive here. Yes. So I'm José Brandão. I'm from Diamond Light Source, and I will follow on talking about what we did with all those proteins we got so far. So initially, back in 2011, Humberto had in Brazil pretty much PNP. It was his PhD work. He had been working with it for a long time. And they already had a normal crystallography type project where they were working on adenosine kinase. And with the OPPF collaboration, we now have all these proteins. So we went from having just the one protein to having a very nice overview, structural overview of the pathway. So 9 unique structures, 60 enzyme ligand complexes, and out of 120 X-ray data sets, which were done on 900 crystals over the last 3 years in about 6 or 7 trips to Diamond, so about 100-something crystals per visit. So it shows how we've been using the synchrotron as well as being, well, myself at least, as well as being staff. And this was definitely very much only possible because of this type of collaboration of using the OPPF as a facility as well. I'll give you one example of one of the proteins, adenosine phosphorylase. It is a protein that was originally thought to exist, but nobody had actually isolated it properly. There was a model that was taken from the human methylthioadenosine phosphorylase for humans, because in humans, this pathway doesn't exist as such, so the adenine is only obtained through the indirect path from adenosine to AMP back to adenine. There is no direct conversion. And for a long time, PNP down here was suggested as being the phosphorylase that could take from adenosine to adenine as well. And once the schistosoma genome was done, the sequence was identified, and it bears 47% identity to the human sequence. And in that OPPF run, the schistosoma adenosine phosphorylase was clone crystallized and biochemically characterized. So the summary of the story is that the schistosoma protein does catalyze both methylthioadenosine, which was the original hypothesis taken from the human protein, but also very much does the adenosine to adenine catalysis. And the very interesting result is that the human form, the MTAP protein, it does not like catalyzing adenosine into adenine. So it's a very nice result in that it is a potential protein target, should we ever pursue that route. One of the type of results we get from doing a fragment or a structural X-ray campaign is this one below here. You have 3 pictures of the active site, with 3 different ligands. First one is tubercidin, which is an adenosine analog, which is an inhibitor for adenosine phosphorylase with MTA and with adenine at the back there. So these are data sets that were taken from mutants of the proteins, of the adenosine phosphorylase binding assays using Raman spectroscopy that can look into how the compounds change during how they are bound to the proteins. And Raman is very good because it looks at a cyclic chemistry. And a very important part of what I aim to do in the future is to find collaborators to do the validation of this target. So, thank you very much.